Hi guys, welcome to part 2. We're going to talk about lipids. So we'll jump right in. What are lipids? So lipids are molecules that are also made of carbon, hydrogen, and oxygen, but they have a lot more hydrogen atoms in them than carbs did. So a couple of their facts about lipids: they are hydrophobic, which means that they do not like to dissolve in water. And you can test this at home. If you have a little bit of vegetable oil or something and you pour it into a glass of water, you'll see that they don't like to mix. They create two separate sections. So the word for that is hydrophobic, meaning that they don't dissolve well in water. They are also used for energy storage, so just like carbs were. So we can use them for energy storage, but they also have a lot of other super important functions. They're used in cell structure, in the membrane of cells. They're used for insulation and protection of nerve cells. They're what allows us to send the impulses through our nerves at a quick pace, so they're super important there. And then one other thing about them is that they can actually store more energy than carbohydrates can, but they're a little bit trickier to use. So the process, if you think back to carbs, where we were talking about this idea of switching back and forth between glycogen and glucose, that process can happen pretty easily. Whereas for lipids, it's a little bit harder. There's more of a process that needs to happen to be able to use them for energy. So we've got 4 types of lipids that we're going to talk about. The first is triglycerides, phospholipids, steroids, and waxes. So we'll start with triglycerides. Triglycerides, as the name suggests, is tri, meaning 3. We've got a glycerol backbone here, and then 3 fatty acid chains that come off of it. So this is the most common type of fat or lipid that is found in plants and animals. And then there's this idea with triglycerides of the fact that the shape of them can kind of change. And so you may have heard of saturated fats, unsaturated fats, trans fats. You may have seen those on nutrition labels. But we're going to talk about what that actually means and what that is and what kind of things in our diets provide each type of fat. So first of all, saturated triglycerides contain only single bonds between atoms. And those are these little lines in here. If it's been a while since you've done chemistry, you might be like, what is that? So you can see in between all of these carbons in the fatty acid chains, we only have one line in between them, and that's called a single bond. So because they're only single bonds, it allows these chains to pack in really tightly together. And so we can stack saturated triglycerides on top of each other really closely. And because of that, they're usually solid at room temperature. They can stack in a bad way. So these types of fats can be responsible for forming plaque in your arteries, and they can increase cholesterol levels that can lead to heart disease or other implications like that. Some examples are things like butter or lard, which are animal fats. And as we know, butter is solid at room temperature, and that is because of those single bonds in the triglyceride molecule. Unsaturated triglycerides have one or more double bonds between carbon atoms. So you can see in green here, we've all of a sudden got 2 lines instead of 1. And you'll notice that at this point, we now have a bend in our fatty acid chain. It's not straight like the other ones. So if you were trying to stack these molecules, it would be harder to do. You can't quite stack them as closely. So that's what makes these types of triglycerides liquids at room temperature, because you can't pack those molecules in as tightly. And this is good. We want to consume this type of lipid because those kinks, or the kind of bend here, they prevent plaque formation. So they're not going to allow these lipids to stack really closely together in our arteries, which is a good thing. So more plant-based fats are typically unsaturated. So things like vegetable oil, olive oil, sunflower oil, those plant-based fats are typically, or oils are typically unsaturated. And then we've got something called trans fats. And so this is something that happens that we can do to food called hydrogenation, which is something that we can do as humans, and that process involves adding or moving hydrogens to try to straighten the line. So because these two, if you look where my mouse is here, because of these two hydrogens close together, that's what causes the bend. So if you take one of those hydrogens off and instead put it on the other side, like here, it'll straighten out that chain. You're going to be able to stack more of these on top of each other, kind of like a saturated fat. So it's this idea, a trans fat is basically an unsaturated fat that has been bent out of shape to resemble a saturated fat. And that's not good. We can't process that very well. So we want to avoid trans fats. Trans fats are bad news bears. And you'll notice that you can look on nutrition labels to see often kind of the typical fatty foods that we think of will have trans fats in them. So you just have to be careful with how many of those you're consuming in your diet. I think we're going to skip that video. It pretty much says exactly what we just went over. So we'll go to the next. Okay, phospholipids. So this is a little bit different from triglycerides because instead of a glycerol head, we now have a phosphate and a glycerol head. So you can see in here we've got glycerol and phosphate. So our head of the lipid is a little bit different, and it actually makes it hydrophilic. So that's the opposite of hydrophobic. Remember, hydrophobic means, and I always think of phobic like a phobia, meaning scared. So hydrophobic means scared of water, whereas hydrophilic means it can dissolve in water. So we've got this head on our lipid that is able to dissolve in water. And then we have hydrophobic fatty acid tails, typically two. We have two tails off of phospholipids. Remember, we had 3 in triglycerides. Usually there's only 2 in phospholipids. These are a super, super essential kind of lipid that we need to have because they make up the cell membranes in all of our cells. Every single cell that we have has this, this cell membrane that is made up of phospholipids. So super, super important. Then we've got steroids, which most of us probably think of something like this picture when you think of steroids, but there's actually natural steroids, which are lipids that we need in our body. So they are usually containing 4 connected carbon rings. And some examples are cholesterol, testosterone, estradiol, cortisol. So if these are ringing a bell, it's because these are hormones. And hormones in our body, which act as messengers to signal different things, are mostly lipids. So those are some types that we have in our bodies. And then last but not least, we have waxes. These can be used by plants and animals as waterproofing agents because they're hydrophobic. Water doesn't stick to them, so they can be really useful for different animals and plants to help create that waterproof effect. Different birds have this on the outside of their wings so that if they're, diving, say, into water for food, as soon as they come out of food, or diving into the water for food, as soon as they come out of the water, that water's not gonna clog their wings. They can dry off really quickly. Also, beeswax is obviously made of wax, different cuticle on leaves as well. So we won't be focusing too much on waxes, but knowing that it's out there and that it's a lipid is important. Okay, this video we will watch. So this is how lipids are digested in our bodies.
Foods, such as meats, dairy products, seeds, nuts, and oils, contain dietary fat. And fat is a common example of a lipid. And in this lesson, you will learn about the unique way lipids, such as fats, are broken down and absorbed out of the digestive tract. Now, a lipid is defined as a fat-like molecule that does not have the ability to dissolve in water. This inability to dissolve in water adds an element of difficulty to fat digestion. Because fat does not like water, it tends to clump together and form large droplets as it moves through your digestive system. By the time fat reaches your small intestine, it has not been digested at all. So dietary fat in the small intestine looks like a fairly large glob of fat. These globs remain until bile that is produced in the liver and stored in the gallbladder mixes with the large fat droplets. Now, bile contains bile salts, which act as an emulsifier of lipids. The term emulsify means to break large fat droplets into smaller droplets. And that is exactly what we see happening here in the small intestine. The bile salts break up and coat the fat to form much finer droplets. These finer droplets have more surface area, and this aids digestion because the fat-digesting enzyme pancreatic lipase can only act on the surface of the fat droplet. The enzymes of the small intestine are responsible for almost all of the fat digestion. When pancreatic lipase acts on the lipid, it breaks it down, which results in free fatty acids and monoglycerides, the two digestive products of lipids. These products are much easier for your small intestine to handle, and they have very little trouble being absorbed out of your digestive tract. Now, absorption takes place through the mucosal lining of the small intestine, and when these products pass through the mucosa, they enter the epithelial cells. Once inside the cells, the free fatty acids and monoglycerides enter the endoplasmic reticulum, which is a system inside the cell whose functions include synthesis and transport of lipids. Here, the digested products are resynthesized into triglycerides. Triglycerides are the major form of fat stored by the body. So an important fact to point out is that fats are reassembled in the epithelial cells of the small intestine. This makes fat absorption different than absorption of proteins and carbohydrates. With protein and carbohydrate absorption, we see their basic units, which are amino acids and monosaccharides, are able to pass through the intestinal epithelial cells without being altered. Now, before the triglycerides actually leave the epithelial cells, they're coated by protein, which results in the formation of chylomicrons. So we see that chylomicrons are small globules composed of protein and lipid. The coating of protein gives the triglyceride a water-soluble coat, and this allows the chylomicron to travel outside of the cell. The newly formed chylomicrons leave the epithelial cell and enter the lymphatic capillaries, which are called lacteals. The lacteals are found in the finger-like projections of the intestinal wall called the villi. The lacteals represent another unique way fats are absorbed, because lipids pass through the lymphatic system before they make their way back to your bloodstream. This differs from protein and carbohydrate digestion because amino acids and monosaccharides do not enter the lymphatic system. Instead, they travel directly into the blood capillaries and then into the hepatic portal vein that leads to the liver. Fats can now travel around your body. They are used to build cell membranes, make the myelin sheaths that coat your nerves, and of course insulate the body with a fatty cushion. Fats can also be used as fuel for your body if there are not enough carbohydrates in your system. And although you need fats for these important body functions, you can have too much of a good thing. For example, a high level of triglycerides in your blood can increase your risk of heart disease.
Okay, so again, we're going to do our own summary of that because it was a little bit complex. So let's break down exactly what you need to know. First of all, in the first section of our small intestine, we have bile that's, being produced by the liver and stored by the gallbladder. That's coming into the small intestine. It's going to break down fat globules into tiny droplets. So that's this first picture here. We've got a lipid globule, and bile salts are going to come in and they surround them, and they are able to break those into smaller little droplets. Enzymes separate the free fatty acids from the monoglycerides. So basically, if you think about the triglyceride molecule, we've got the glycerol head and the three fatty acid chains. So we're breaking those apart using enzymes. So then those are going into these cells. So hopefully you can see where my mouse is here, but in the small intestine, instead of just passing right through, these separate pieces, the glycerol and the free fatty acids, they are going to go right into these cells, and then they're going to be reformed. So they're taken apart and then they're kind of put right back together. And then triglycerides are absorbed into the lymphatic vessels and then the bloodstream. So that is another difference there, is instead of going right into the bloodstream, we're going into the lymphatic system first. And then the last important point is that if needed, glycerol or a monoglyceride can be converted into glucose to provide energy. And like I said earlier, it's a bit more of a kind of complex process to do this, but it is possible. We can burn fat for energy if need be. And then lastly, where do lipids come from? They come from our meat products. They come from our dairy products, cheeses and milks. We can get some of our not so good lipids from greasy food. And then cooking with any kind of oil is also a way to get lipids. So that's it for lipids. Come join for part 3, proteins, up next.